So of course, we develop our own classifier, which again will not be the best, but at least an attempt. So we try to learn from multiple references at the same time. Yeah. So again, there is a preprint. You can check the details here. I will just explain it very briefly. So if we have two data sets, one is annotated at 3 levels, so 1, 2, 3, and then this is A, B, C, D. So think about this. These are like, I don't know, this data set came with T cells only, and this one gave you CD4 and CD8 T cells. So what we do is that we train one classifier on this data, we train another classifier on this data. Doesn't matter which classifier we use, SVM. As you see, I'm a big fan, but it can be anything at this point. And then we cross them with each other. So we use the classifier trained on dataset 2 to label dataset 1 and the other way around. What will happen here is the classifier will say A and B are both 1, right? That's what it knows. And it will say that 1 is actually half A and half B. And we use this information to reconstruct this classification tree that we now know that this sample has 1, 2, 3, and 1 has 2 subpopulations, A and B. Yeah. And later on, we can, if we get a third data set, we can, that's unlabeled, we can use this tree to classify the data that we get, and then add it back to the tree as well. Yeah. So here is a bit an example on PBMC data. So these are 3 data sets that we looked at. And these are the populations that that are part of these data sets. And you see, for instance, I don't know, B cells are present in all data sets. But if we look here, for instance, at CD4 T cells, they are annotated at this level in 2 of the data sets. But in the third data set, you have subpopulations of CD4. Yeah, this is kind of the tree that you expect. And this is the tree that we constructed. So again, as I said, B cells are very easy. So you can easily make the define that. NK cells were also quite okay, but we make a lot of mistakes and also bad mistakes. So here, for instance, NK cells from one data set, we group them as subpopulations of CD8. For instance, here for the MK cells, we actually have 2 subpopulations. And at some point, we don't know if this is correct or not, right? They are both MK cells, so they should be together. If you look at the UMAP of the integrated data, you see that they are slightly different. Like one of them is lying to the right, one of them is to the left of this cluster. So they might be slightly different, but we really don't know if this is just a batch effect or actual biology. So once we have this tree, it's not perfect, but we can still use it for classification. And that's what I'm showing you here. So these are the true labels of a 4th data set that was never used in training. And these are the predicted labels. And things are more or less along the diagonal, so you think it's actually quite okay. There is something here. You see the CD16 MC cells and MDC are totally flipped. So whatever is MDC, we predict to be MC, and whatever is MC, we predict to be DC. This is actually a problem in the original labeling of the data we figured out later. So this is not the classifier making a mistake. This is just a wrong label of the data. Yeah, we think this is a nice way to learn if you have multiple atlases and you still don't have a very good established about what to expect in the data. Of course, PBMC is not the right example where you would use this because we know quite a bit about hematopoietic differentiation, but that's why we use it as well. So we can also judge a little bit how well we're doing.